So DNA as an acidic substance present in the nucleus, it was first identified by Friedrich Miescher. He named it nuclein. So Friedrich Miescher was the first one to identify that there is some acidic substance which is present in the nucleus, and he named it nuclein. But then due to technical limitation, isolating the entire DNA molecule was not possible at that time. So because of that, the structure of DNA, it was a mystery for a very long period of time. They just know that there is an acidic substance called DNA is present in the nucleus, but then no one knew about its structure. But only in the year 1953, James Watson and Francis Crick, they proposed a very simple but famous double helix model for the structure of DNA. This model is very important and it was like a turning point in the structure of DNA identification. So this proposition was based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, and it was also based on the observation which was made by Erwin Chargaff that for a double-stranded DNA molecule, the ratio between adenine and thymine and guanine and cytosine are constant and it equals to 1. So one of the hallmark or the important point of the double helix model for the structure of DNA is the base pairing between the two strands of polynucleotide chain. So now let us see about the double helix structure of DNA. Okay, so it is made up of two polynucleotide chains. Okay, so here is one chain, and then here you have another chain. So it is made up of two polynucleotide chain, and if you consider the backbone, it is made up of sugar and phosphate. This backbone, it is made up of sugar and phosphate as we have already seen, and this bases will project inside from the backbone. And another thing is that next point is that the two chains have antiparallel polarity. That is, if one chain has the polarity phi dash to 3 dash, okay, if one chain has the polarity phi dash to 3 dash, the opposite strand will have the polarity 3 dash to phi dash. So we have already seen what does this polarity means, because in a single strand of polynucleotide chain, one end it will have a free phosphate group which is referred to as phi dash N, because phosphate groups are always attached to the fifth position in a pentose sugar, and in the other side it will be having a free OH group which is referred to as 3 dash end of a polynucleotide chain, because the OH groups are always attached to the 3 dash position in a pentose sugar. So next point is that the bases in two strands, they are paired through hydrogen bonds. Here you can see that the bases are bonded. That is why it is possible to have the compatibility of two strands together, right? So the bases in two strands are paired through hydrogen bonds, forming base pairs. So adenine, it will form two hydrogen bonds with thymine. Here you can see it forms two hydrogen bonds with thymine, right? And guanine, it is bonded with cytosine with 3 hydrogen bond. It is bonded with 3 hydrogen bond, and this is a rule, okay? Adenine and thymine will always be bonded together with 2 hydrogen bond, and guanine and cytosine will always be bonded together with 3 hydrogen bonds. This is in case of DNA. So this is an advantage, because if you know what is the bases present in one strand, then you can enumerate the bases which are present in the opposite strand. So because of this, a purine always comes opposite to that of a pyrimidine, because adenine and guanine, they are purines. So opposite to that I have thymines and cytosines which are pyrimidines. The next point is that the two chains, okay, the two opposite strands of the polynucleotide chains, they are coiled in a right-handed fashion. You can see that it is in a right-handed fashion, in a helical fashion, right? So the pitch of the helix is 3.4 nanometer, Okay, this pitch. The pitch of this helix is 3.4 nanometer, and approximately there are 10 base pairs in each pitch. Okay, in this region, if there are approximately 10 base pairs, then the distance between each base pair, the distance between each base pair will be nothing but 3.4 divided by 10, which will be equal to 0.34 nanometer. Right, it will be equal to 0.34 nanometer. So consequently, the distance between each base pair in a helix will be 0.34 nanometer. And then another thing is that the last point is that the plane of one base pair stacks over the other. It lies over the other in the double helix structure. So in addition to hydrogen bonds, this will confer stability to this helical structure. Now coming to the central dogma of molecular biology. So we know that this double helix structure proposition made the structure of DNA to be understood in a very easy manner, right? So it became extremely revolutionary. So soon after this proposition of the double helix structure, Francis Crick, he proposed the central dogma in molecular biology. So the central dogma in molecular biology, it states that the genetic information, it flows from DNA to RNA and then to protein. Okay, the genetic information flows from DNA to mRNA and then to protein. In other words, we can say that DNA will give rise to RNA, and from RNA proteins are formed. So a DNA will again give rise to DNA, that is, more DNA will be replicated by the process of replication, right? And then DNA will give rise to mRNA by the process of transcription. And then mRNA will give rise to protein by the process of translation, about which you will be studying.